muscle fiber generates tension during the action of actin and myosin cross-bridge cycling. While under tension, the muscle may lengthen, shorten, or remain the same. Although the term contraction implies shortening, when referring to the muscular system, it means muscle fibers generating tension with the help of motor neurons. Voluntary muscle contraction is controlled by the central nervous system. The brain sends signals, in the form of action potentials, through the nervous system to the motor neuron that innervates several muscle fibers. In the case of some reflexes, the signal to contract can originate in the spinal cord through a feedback loop with the gray matter. Involuntary muscles such as the heart or smooth muscles in the gut and vascular system contract as a result of non-conscious brain activity or stimuli proceeding in the body to the muscle itself. Classification: skeletal muscle contractions can be broadly separated into twitch and tetanic contractions. In a twitch contraction, a short burst of stimulation causes the muscle to contract, but the duration is so short that the muscle begins relaxing even before reaching peak force. The shape of the graph of force versus time in a twitch contraction can give information about the relative rates of calcium release and reuptake from the sarcoplasmic reticulum. If the stimulation is long enough, the muscle reaches peak force and plateaus at this level, resulting in a tetanic contraction. If the stimulation is not intense enough, force will oscillate during the plateau and be submaximal, but with sufficient stimulation, there will be a constant force level until stimulation stops. Voluntary muscular contractions can be further classified according to either length changes or force levels. In spite of the fact that the muscle actually shortens only in concentric contractions, all activations are typically referred to as contractions. In concentric contraction, the force generated is sufficient to overcome the resistance, and the muscle shortens as it contracts. This is what most people think of as a muscle contraction. In eccentric contraction, the force generated is insufficient to overcome the external load on the muscle and the muscle fibers lengthen as they contract. An eccentric contraction is used as a means of decelerating a body part or object, or lowering a load gently rather than letting it drop. In isometric contraction, the muscle remains the same length. An example would be holding an object up without moving it. The muscular force precisely matches the load, and no movement results. In isotonic contraction, the tension in the muscle remains constant despite a change in muscle length. This can occur only when a muscle's maximal force of contraction exceeds the total load on the muscle. In isovelocity contraction, the muscle contraction velocity remains constant, while force is allowed to vary. True isovelocity contractions are rare in the body, and are primarily an analysis method used in experiments on isolated muscles that have been dissected out of the organism. In reality, muscles rarely perform under any sort of constant force, velocity, or speed, but these contractions are useful for understanding overall muscle properties present in more complex contractions that occur in vivo. Cyclic in vivo contractions can be modeled using work loops. Concentric contraction, eccentric contraction or shortening contraction is a type of muscle contraction in which the muscles shorten while generating force. This occurs when the force generated by the muscle exceeds the load opposing its contraction. During a concentric contraction, a muscle is stimulated to contract according to the sliding filament mechanism. This occurs throughout the length of the muscle, generating a force at the musculo-tendinous junction, causing the muscle to shorten and changing the angle of the joint. In relation to the elbow, a concentric contraction of the biceps would cause the arm to bend at the elbow as the hand moved from the leg to the shoulder. A concentric contraction of the triceps would change the angle of the joint in the opposite direction, straightening the arm and moving the hand towards the leg. Eccentric contraction: during an eccentric contraction, the muscle elongates while under tension due to an opposing force greater than the muscle generates. Rather than working to pull a joint in the direction of the muscle contraction, the muscle acts to decelerate the joint at the end of a movement or otherwise control the repositioning of a load. This can occur involuntarily or voluntarily. Over the short term, strength training involving both eccentric and concentric contractions appear to increase muscular strength more than training with concentric contractions alone. However, Exercise-induced muscle damage is also greater during lengthening contractions. During an eccentric contraction of the biceps muscle, the elbow starts the movement while bent and then straightens as the hand moves away from the shoulder. During an eccentric contraction of the triceps muscle, the elbow starts the movement straight and then bends as the hand moves towards the shoulder. Desmin, titin, and other Z-line proteins are involved in eccentric contractions, but their mechanism is poorly understood in comparison to cross-bridge cycling and concentric contractions. Though the muscle is doing a negative amount of mechanical work, chemical energy is nevertheless consumed, although less than would be consumed during a concentric contraction of the same force. For example, one expends more energy going up a flight of stairs than going down the same flight. Muscles undergoing heavy eccentric loading suffer greater damage when overloaded as compared to concentric loading. When eccentric contractions are used in weight training, they are normally called negatives. During a concentric contraction, muscle fibers slide across each other, pulling the Z-lines together. During an eccentric contraction, the filaments slide past each other the opposite way, though the actual movement of the myosin heads during an eccentric contraction is not known. Exercise featuring a heavy eccentric load can actually support a greater weight and also results in greater muscular damage and delayed onset muscle soreness 1 to 2 days after training. Exercise that incorporates both eccentric and concentric muscular contractions can produce greater gains in strength than concentric contractions alone. While unaccustomed heavy eccentric contractions can easily lead to overtraining, moderate training may confer protection against injury. Eccentric contractions in movement. Eccentric contractions normally occur as a braking force in opposition to a concentric contraction to protect joints from damage. During virtually any routine movement, eccentric contractions assist in keeping motions smooth, but can also slow rapid movements such as a punch or throw. Part of training for rapid movements such as pitching during baseball involves reducing eccentric braking, allowing a greater power to be developed throughout the movement. Eccentric contractions are being researched for their ability to speed rehabilitation of weak or injured tendons. Achilles tendinitis has been shown to benefit from high-load eccentric contractions. Isometric contraction. An isometric contraction of a muscle generates force without changing length. An example can be found when the muscles of the hand and forearm grip an object. The joints of the hand do not move, but muscles generate sufficient force to prevent the object from being dropped. Physiology. For voluntary muscles, all contraction occurs as a result of conscious effort originating in the brain. The brain sends signals, in the form of action potentials, through the nervous system to the motor neuron that innervates several muscle fibers. In the case of some reflexes, the signal to contract can originate in the spinal cord through a feedback loop with the gray matter. Involuntary muscles such as the heart or smooth muscles in the gut and vascular system contract as a result of non-conscious brain activity or stimuli endogenous to the muscle itself. Other actions such as locomotion, breathing, and chewing have a reflex aspect to them. The contractions can be initiated both consciously or unconsciously. There are 3 general types of muscle tissues: skeletal muscle responsible for movement, cardiac muscle responsible for pumping blood, smooth muscle responsible for sustained contractions in the vascular system, gastrointestinal tract, and other areas in the body. Skeletal and cardiac muscles are called striated muscle because of their striped appearance under a microscope, which is due to the highly organized alternating pattern of A band and I band. While nerve impulse profiles are, for the most part, always the same, skeletal muscles are able to produce varying levels of contractile force. This phenomenon can be best explained by force summation. Force summation describes the addition of individual twitch contractions to increase the intensity of overall muscle contraction. This can be achieved in 2 ways, by increasing the number and size of contractile units simultaneously, called multiple fiber summation, and by increasing the frequency at which action potentials are sent to muscle fibers, called frequency summation. Multiple fiber summation when a weak signal is sent by the CNS to contract a muscle, the smaller motor units, being more excitable than the larger ones, are stimulated first. As the strength of the signal increases, more motor units are excited in addition to larger ones, with the largest motor units having as much as 50 times the contractile strength as the smaller ones. As more and larger motor units are activated, the force of muscle contraction becomes progressively stronger. A concept known as the size principle allows for a gradation of muscle force during weak contraction to occur in small steps, which then become progressively larger when greater amounts of force are required. Frequency summation for skeletal muscles, the force exerted by the muscle is controlled by varying the frequency at which action potentials are sent to muscle fibers. Action potentials do not arrive at muscles synchronously, and, during a contraction, some fraction of the fibers in the muscle will be firing at any given time. In a typical circumstance, when a human is exerting a muscle as hard as she is consciously able, roughly 1/3 of the fibers in that muscle will be firing at once, though this ratio can be affected by various physiological and psychological factors. This low level of contraction is a protective mechanism to prevent avulsion of the tendon. The force generated by a 95% contraction of all fibers is sufficient to damage the body. Skeletal muscle. The sliding filament theory describes a process used by muscles to contract. It is a cycle of repetitive events that cause a thin filament to slide over a thick filament and generate tension in the muscle. It was independently developed by Andrew F. Huxley and Rolf Niederschak and by Hugh Huxley and Gene Hansen in 1954. An action potential originating in the CNS reaches an alpha motor neuron, which then transmits an action potential down its own axon. The action potential propagates by activating voltage-gated sodium channels along the axon toward the neuromuscular junction. When it reaches the junction, it causes a calcium ion influx through the voltage-gated calcium channels. The Ca2+ influx causes vesicles containing the neurotransmitter acetylcholine to fuse with a plasma membrane, releasing acetylcholine out into the extracellular space between the motor neuron terminal and the neuromuscular junction of the skeletal muscle fiber. The acetylcholine diffuses across the synapse and binds to and activates nicotinic acetylcholine receptors on the neuromuscular junction. Activation of the nicotinic receptor opens its intrinsic sodium-potassium channel, causing sodium to rush in and potassium to trickle out. Because the channel is more permeable to sodium, the charge difference between internal and external surfaces of the muscle fiber membrane becomes less negative, triggering an action potential. The action potential spreads through the muscle fiber's network of T-tubules, depolarizing the inner portion of the muscle fiber. The depolarization activates L-type voltage-dependent calcium channels in the T-tubule membrane, which are in close proximity to calcium release channels in the adjacent sarcoplasmic reticulum. Activated voltage-gated calcium channels physically interact with calcium release channels to activate them, causing the sarcoplasmic reticulum to release calcium. The calcium binds to the troponin C present on the actin containing thin filaments of the myofibrils. The troponin then allostrically modulates the tropomyosin. Under normal circumstances, the tropomyosin sterically obstructs binding sites for myosin on the thin filament. Once calcium binds to the troponin C and causes an allostric change in the troponin protein, troponin T allows tropomyosin to move, unblocking the binding sites. Myosin binds to the newly uncovered binding sites on the thin filament. Myosin is now bound to actin in the strong binding state. The release of ADP and inorganic phosphate are tightly coupled to the power stroke. This will pull the Z bands towards each other, thus shortening the sarcomere and the I band. ATP binds to myosin, allowing it to release actin and be in the weak binding state. The myosin then hydrolyzes the ATP and uses the energy to move into the cocked-back conformation. In general, evidence indicates that each skeletal muscle myosin head moves 10-12 nm each power stroke, however there is also evidence of variations that appear specific to the myosin isoform. Steps 9 and 10 repeat as long as ATP is available and calcium is freely bound within the thin filaments. While the above steps are occurring, calcium is actively pumped back into the sarcoplasmic reticulum. When calcium is no longer present on the thin filament, the tropomyosin changes conformation back to its previous state so as to block the binding sites again. The myosin ceases binding to the thin filament, and the contractions cease. The calcium ions leave the troponin molecule in order to maintain the calcium ion concentration in the sarcoplasm. The active pumping of calcium ions into the sarcoplasmic reticulum creates a deficiency in the fluid around the myofibrils. This causes the removal of calcium ions from the troponin. Thus, the tropomyosin-troponin complex again covers the binding sites on the actin filaments and contraction ceases. Physiologically, this contraction is not uniform across the sarcomere. The central position of the thick filaments becomes unstable and can shift during contraction. However, the actions of elastic proteins such as titin are hypothesized to maintain uniform tension across the sarcomere and pull the thick filament into a central position. Smooth muscle. The interaction of sliding actin and myosin filaments is similar in that of smooth muscle. There are differences in the proteins involved in contraction in vertebrate smooth muscle compared to cardiac and skeletal muscle. Smooth muscle does not contain troponin, but does contain the thin filament protein tropomyosin, and other notable proteins are urocordsman and calponin. Contractions are initiated by the calcium-activated phosphorylation of myosin rather than calcium binding to troponin. Contractions in vertebrate smooth muscle are initiated by agents that increase intracellular calcium. This is a process of depolarizing the sarcolemma and extracellular calcium entering through L-type calcium channels, and intracellular calcium release predominantly from the sarcoplasmic reticulum. Calcium release from the sarcoplasmic reticulum is from ryanodine receptor channels by a redox process and inositol triphosphate receptor channels by the second messenger inositol triphosphate. The intracellular calcium binds with carmodulin, which then binds and activates myosin light chain kinase. The calcium carmodulin myosin light chain kinase complex phosphorylates myosin on the 20 kilodalton myosin light chains on amino acid residue serine 19, initiating contraction and activating the myosin at pace. The phosphorylation of cordsman and calponin by various kinases is suspected to play a role in smooth muscle contraction. Phosphorylation of the 20 kDa myosin light chains correlates well with the shortening velocity of smooth muscle. During this period, there is a rapid burst of energy utilization as measured by oxygen consumption. Within a few minutes of initiation, the calcium level markedly decreases, the 20 kDa myosin light chains phosphorylation decreases, and energy utilization decreases. However, force in tonic smooth muscle is maintained. During contraction of muscle, rapidly cycling cross bridges form between activated actin and phosphorylated myosin, generating force. It is hypothesized that the maintenance of force results from dephosphorylated latch bridges that slowly cycle and maintain force. A number of kinases such as Rho kinase, ZIP kinase, and protein kinase C are believed to participate in the sustained phase of contraction, and calcium flux may be significant. Invertebrate smooth muscles. In invertebrate smooth muscle, contraction is initiated with calcium directly binding to myosin and then rapidly cycling cross bridges generating force. Similar to vertebrate tonic smooth muscle, there is a low calcium and low energy utilization catch phase. This sustained phase or catch phase has been attributed to a catch protein that is similar to myosin light chain kinase and titin, known as twitchin. History. Excitation. Contraction coupling was a term coined in 1952 to describe the physiological process of converting an electrical stimulus to a mechanical response. This process is fundamental to muscle physiology, whereby the electrical stimulus is usually an action potential and the mechanical response is contraction. EC coupling can be dysregulated in many diseases. Though EC coupling has been known for over half a century, it is still an active area of biomedical research. The general scheme is that an action potential arrives to depolarize the cell membrane. By mechanisms specific to the muscle type, this depolarization results in an increase in cytosolic calcium that is called a calcium transient. This increase in calcium activates calcium-sensitive contractile proteins that then use ATP to cause cell shortening. the mechanism for muscle contraction evaded scientists for years and requires continued research and updating. The sliding filament theory described a process used by muscles to contract and is now the accepted theory for muscle contraction. It is a cycle of repetitive events that cause a thin filament to slide over a thick filament and generate tension in the muscle. It was independently developed by Andrew F. Huxley and Rolf Niederschak and by Hugh Huxley and Gene Hansen in 1954. Force length and force velocity relationships. Unlike mechanical systems such as motors, the force a muscle can generate depends upon both the length and shortening velocity of the muscle. Force-length relationship, also called the length-tension curve, relates the strength of an isometric contraction to the length of the muscle at which the contraction occurs. Muscles operate with greatest active force when close to an ideal length. When stretched or shortened beyond this, the maximum active force generated decreases. This decrease is minimal for small deviations, but the force drops off rapidly as the length deviates further from the ideal. As a result, in most biological systems, the range of muscle contraction will remain on the peak of the length-tension curve, in order to maximize contraction force. Due to the presence of elastic proteins within a muscle, as the muscle is stretched beyond a given length, there is an entirely passive force, which opposes lengthening. Combined together, we see a strong resistance to lengthening an active muscle far beyond the peak of active force. Force-velocity relationship, the speed at which a muscle changes length also affects the force it can generate. Force declines in a hyperbolic fashion relative to the isometric force as the shortening velocity increases, eventually reaching zero at some maximum velocity. The reverse holds true for when the muscle is stretched. Force increases above isometric maximum, until finally reaching an absolute maximum. This has strong implications for the rate at which muscles can perform mechanical work. Since power is equal to force times velocity, the muscle generates no power at either isometric force or maximal velocity. Instead, the optimal shortening velocity for power generation is approximately 1/3 of maximum shortening velocity. These two fundamental properties of muscle have numerous biomechanical consequences, including limiting running speed, strength, and jumping distance and height. See also, cramp, dystonia, exercise physiology, fasciculation, Hill's muscle model, hypnic jerk, in vitro muscle testing, myoclonus, spasm, supination, uterine contraction. References. External links: sliding filament model of muscle contraction, physiology of skeletal muscle contraction. Medrome, animation, myofilament contraction.